So we're going to talk about glowing mushrooms tonight, bioluminescent and fluorescent mushrooms. But I'll say a couple things first about a couple other things. And you can hit the lights in like a minute or whatever. But I've been doing a lot of identifications on iNaturalist. But there's too much rain in California now, and I can't identify everything in the whole state. That'll be a full-time job. But if you find some stuff and no one's identifying it, just message me your username and I can flip through your account and put names on stuff that you found. Also, if you have any questions about anything I'm saying tonight, you can feel free just to shout out the questions. I'll take questions at the end, but it's probably best if you just ask things right when you think about it. And I think the talks are better that way. When people just interrupt me in mid-sentence, that's totally fine. Also, you can go ahead and hit the lights. If some of these photos look really cool, it's because I've been using a technique called photo stacking. And I wanted to show you really quick how that works. So what I do is I set up my camera and I open the aperture up all the way and turn the ISO all the way down. And then I tell it to take between 40 and 100 pictures. And then I load those onto the computer and combine all those photos into one photo that's really good quality. And the reason it's so cool is it kind of bends the laws of physics because I can get infinite depth of field, and then when the aperture is wide open, then the resolution is just incredible. So I'm going to show you really quick how it works. Here's some pictures I took a few days ago, and this is Xylaria hypoxylon. And so I just selected a whole bunch of pictures, and if I throw those into Helicon, so Helicon is one program that you can use, but there's other ones. Looks like I should probably close this, start it over. There's Combine ZP, which is free, and Helicon costs some money but has a free 30-day trial, and then Zareen Stacker is another one that people use. Anyways, you just select all the photos that you want to combine into one photo and drag it over into Helicon, and then you hit go. And if you look at this, it's just combining them right now. And you can see the image come in on the right. And now I can save this, but I don't know how to zoom in without my mouse. But if I did zoom in, you can see that the resolution on this is just incredible. It's super fine detail, just because the aperture is all the way open. So I use that with every photo I take now. So, it used to be that I would set up the camera and turn the aperture to f/22 and then, take a picture that way. But now I just leave the aperture wide open. So it takes about 10 seconds to take the picture and then another 30 seconds on the computer. So it's a little bit more time, but it's totally worth it. So we'll talk about glowing mushrooms. And let's see, exit. So there's two kinds of glowing mushrooms. There are luminescent mushrooms and fluorescent mushrooms. And we'll talk about both of them. The bioluminescent, or maybe the bio is redundant, and we just call them luminescent. Bioluminescent organisms make their own light, so in complete darkness, they glow. And then fluorescent organisms turn ultraviolet light into visible light, so you shine a black light on them, and then they glow in the black light. And then phosphorescent is the other word that you hear sometimes. And phosphorescent things, you shine light on them, and then they glow for a few seconds or maybe a few hours afterwards. So here is a phosphorescent t-shirt. So I just shined a black light on it to charge it up. And then this t-shirt's kind of cool, though. I'll wake up in the middle of the night and look over, and the mushrooms are still glowing. So there's no phosphorescent mushrooms. I do sometimes find phosphorescent rocks, I shine the ultraviolet on the rock, and then when I shine it away, the rock glows for a few seconds. But no, nothing alive is phosphorescent. And then fluorescence, you shine the black light on it, and it converts the higher energy, more short wavelength light to lower energy, longer wavelength light. And so in fluorescent mushrooms, you can get a ton of different colors. You can see, here's a puffball I found at Telluride. And the outside is fluorescent blue, and the inside is fluorescent green. So just cutting them in half made them look really cool. But you can get just about every color in the rainbow with fluorescence. And then bioluminescence is where they make their own light, and there's only one color of bioluminescence, and that is 530 nanometers, which happens to be a green. So scientists express color in a number of nanometers. That's just the wavelength of the light. So all of the glowing mushrooms where you don't have to shine a black light on them, they always glow green. So first I'll talk about the bioluminescent mushrooms. This is one that you have around here, and this is armillaria. So armillaria, it looks like, the piece of wood looks like this, and you can see these zones of inhibition there, these black zones. And so I always take the exact same picture on a tripod in the complete darkness and then with light so you can see what it is that is glowing. So it's really just that inside part there. And so these are honey mushrooms, and the mushroom itself doesn't glow at all. It's only the mycelium that's glowing. So this stump, somebody found it at Salt Point State Park, and the whole stump was glowing, and they just brought me a piece of it. And so I took a little bit and I sequenced the DNA so I know what kind of honey mushroom it is. It's Armillaria centipina. But those are pretty cool. And then the most common luminescent mushroom in Southern California is the jack-o'-lantern. So this one's Omphalotus olivescens, and it's super common, and they're really big. And they seem to glow brightest when they're kind of like large and messed up. So here's some. These, I just found them in the back of a truck, and I'm like, hey, can I have those? And he's like, yeah. So I brought him over to Counterculture Labs, so that's a biohacker space in the East Bay. And it's sort of like a science club where you pay a few bucks per month, and then you get 24/7 access to the laboratory. And it's kind of cool because they have a million chemicals there, and you can do DNA sequencing and macroscopy and all that kind of stuff. I think there's one around here in Los Angeles, too. But in any case, I just brought this mushroom into the back room and did like a, 2-minute exposure or something. But these mushrooms are really big. Another really common one is Panella stypticus, and this is a really common one that people grow. So here is just a grow bag. People grow it on sawdust. And one time, my friend Blue Helix gave me a sawdust bag of Panella stypticus, and he just autoclaved it and then injected the liquid culture that he ordered from Sporeworks. And that thing was glowing for about 18 months. And my room had no windows at that time because I was living in this artist collective in San Francisco. So I would just like have this sawdust block, and when I woke up in the middle of the day or night, didn't matter because there was no windows, I could like look up and see this glowing block of mycelium and kind of like orient myself and know which way to walk out the door.
You can make an exit sign.
You definitely could. So the mycelium does glow in this, but then when it fruits, it glows about 10 times brighter. So usually when people are growing glowing mushrooms, it's this one. And then here's the same thing, Panella stypticus, that I found in Michigan. So this one, kind of looks like a polypore. It's actually really closely related to Mycena. And
speaking of Michigan, Julia Denoke, she's a herbalist, indigenous from Anishinaabe. In her book, she talks about the herbalists that she was apprenticed to kept bioluminescent wood around her door so that people could find her in the night.
that's awesome.
Why is that white stuff on it? Yeah.
What's that?
Why does it have white stuff on it?
I guess it's just like mycelium. I don't know. So this one you can see is definitely focus stacked because you get like really good resolution all the way back there. And here's like a 1-second exposure. So I was just holding this Panella stypticus log. And then they say that it doesn't glow on the West Coast, but here's some that I found in Sonoma County. Definitely glowing. Here's another one from Somacamp. This one is Mycena roritus. So you see it's got the super slimy stem. And this one I took it in the middle of the day, but there was, I just went into the bathroom, and the bathroom had no windows, but there was like light coming in from under the door. So I put a towel under the door like a stoner, but there was still light coming in. There was still light coming in from like the side crack of the door, so I kind of like got, you can kind of like see the mushroom, but you can also see it glowing at the same time. And then this one is a petri dish, and it has honey mushroom mycelium in it. So this is Armillaria cinepina, the heart singer, collected at Salt Point State Park. And you can see that like where the mycelium surfaces out of the agar is where it's glowing most. And honey mushroom always does this, makes these crazy tentacles on agar. No. Here's another one that people don't usually think of as a bioluminescent mushroom. This one is Mycena haematopus. So super common mushroom, and you can see it's just glowing very little bit, like just the stem bases. And also like where insects bite into it, it'll kind of glow a little bit there too. And then here's a Mycena from Gainesville, Florida. And this Mycena, like the mushroom doesn't glow at all, but all these leaves that have the mycelium on it are glowing. So here's a muscadine grape leaf, and it's got all this awesome rhizomorphic mycelium on it that's glowing it. So it's from that Mycena. And then here's some I found in Ecuador. This is probably a Geronema. So a kind of cool thing with decurrent gills, the whole thing glows. And then this is from Oaxaca. This is Mycena lumina. And so this I found at the base of a landslide at about 3 in the morning. And so super dangerous walking around these landslides in the middle of the night, but totally worth it. And you can see this glowing. These are really bright. You can see this glowing from a long ways away. And then here's another one. This one's from Veracruz, Mycena lux follicula. And all of these had no name when I found them, but they're all named now. And this one's really cool because the stem is like really bright. So you're walking through the woods at night and you see like a vertical line, a whole bunch of vertical lines in the woods, and it's all the stems of all these things. And then here's my favorite one, Mycena globulispora. And globulispora, it's super dim, but it's the edges of the gills that are glowing. And this one was recently described from Brazil, though the people who named it didn't realize that it was a glowing mushroom. But this one, it was really dim. It was so dim that I could just barely notice that it was glowing. So I set the camera up, and then I just opened the shutter and went to sleep. And after 40 minutes, the battery ran out, and it saved the photo. And so it's a 40-minute exposure. So that was like F8 ISO 800 or something. Here's Mycena fulguris, also from Veracruz. So I think Veracruz, Mexico is like in the southeast. I think it has more glowing mushrooms than anywhere else in the world. It also has more species of psilocybe than anywhere else. And those things kind of go together, like these glowing mycenas, like the same habitat as the psilocybe, those cloud forest habitats. And so I found like 26 species that glow there, and there's a lot more to be discovered. Yes, there's definitely a reason. I have no idea what it is. No, I have no idea why they're always green. I guess, these glowing mushrooms, they glow because of a reaction between luciferase, the enzyme, and luciferin, and oxygen. And that particular enzyme, it just emits green photons, so that's just which chemicals they happen to have. But I don't know if there's any reason why it's green or if other colors of light would serve it better. And I also don't know if they do anything for the mushroom or if it's just, maybe they're like getting rid of excess oxygen or chemicals that they don't want.
Are there studies or research on other countries that have glowing mushrooms? You mentioned that Veracruz has the highest population, but is there like India or South Asia or something like that?
There's a little bit less studies there. If you read all of the scientific literature, it says that Brazil is the best place in the world for glowing mushrooms because I found 14 different glowing species in one little province of Brazil. So I found like 10 more than that in Veracruz, Mexico. So I think that's the best place. But there's a lot of places that are not studied very well. And I'm sure if you go to like China, you would find a ton of these. This is the first one that I ever found. This is Mycena nebula. And this one's really cool because it only grows on the bark of liquid amber trees. And there can be thousands of them. So it can look like a nebula of stars. Yeah, I set up a tripod. I do this all in the middle of the night, or I just collect a bunch of mushrooms, throw them in the tackle box, and then bring them into a dark room with no windows. But most of these I took at night. And so I set up a tripod, and what I do is first I take a photo with the light. So I'll usually use like my cell phone flashlight, so I'll open the shutter for about 3 seconds, and then I'll move my cell phone in an arc over the lens. That way the light's coming from all directions and I don't have a harsh shadow. And then if that exposure looks good, then I just do like 30 seconds at ISO 200,000. And so ISO 200,000 makes it super sensitive, but it's like really grainy at the same time. But based on how the ISO 200,000 one looks, I will turn it down to something reasonable, like ISO 800, and then leave the shutter open for like 3 to 5 minutes. And then that's the final one that I use. But I end up with 2 photos where one of them has light and one of them is in the dark. And then I load them both into Photoshop and I align them like in different layers. And that way I can crop them both exactly the same and then save them both so I can just like throw them into a PowerPoint and flip between them. This one is Mycena lux arboricola, which was described from Brazil. And the people who found it only found like 2 tiny glowing mushrooms, so this is a way better collection. But this one was from a coffee plantation. From an oak tree that was growing in the middle of a coffee plantation. And the stem base glows. And besides the stem base, also like wherever a bug was biting on the cap, that glows too.
Do you think the glowing could be attracting bugs?
It definitely could be. There's a recent study that says that that's not the reason that they glow, but I don't believe it. I think it's very likely that it is what it's doing. Oh, it's hard to tell. I mean, it's hard to ask a bug if they're tripping or not. But definitely, if it's the middle of the night and you're a bug and you're walking around, like a glowing thing is going to be super interesting. You're going to be, attracted toward it. So maybe you spread the spores that way. It's also interesting to note that all of the glowing mushrooms are wood lovers or just saprotrophs. So none of them are mycorrhizal. So maybe that's like bringing it to new substrate or something. This one is Mycena stylobates, and the mushroom doesn't glow, but the mycelium glows really well. So this is a beech leaf, and if you look at the parts that are glowing, they're the parts that are bleached out, and then the darker parts are not glowing. So you can see that the fungus that's eating the leaf is also bleaching it out and eating the color. And these glowing leaves are really cool. Like when I walk around southern Mexico at night, sometimes you'll see like 100,000 of these glowing leaves in your field of view, and it just looks like a whole glowing ocean or something. And then what I'll do is I will collect all the glowing leaves and throw them in a bag, and then after about half an hour of that, I'll turn the flashlight on and then go through my bag of glowing leaves, and like 1 in 100 of them will have mushrooms coming out of it. And so that's how I find these glowing mushrooms. One of the ways. This one doesn't have a name yet, but it will have a name in a couple of years. This will be Mycena lux minor. And so it's growing on a liquid amber seed pod. And the mushroom doesn't glow, but the primordia glows really bright. And also the mycelium glows. And it's very tiny. Like these caps are like 1 millimeter across. Because my friend is naming them all. So I just give him the collections and then he publishes them. Here's one I found this for the first time in Hidalgo, Mexico in 2013. And this one, I was like camping out in the rain and I told my friend that I was going to go find glowing mushrooms and he just laughed at me. And so I like step away from the fire and it's really cold and rainy. And after like 50 feet, I step in a deep puddle. And I was like, man, this is a terrible idea. But I kept walking, and after like a 50 more feet, I saw like really bright glowing mushrooms. And so this one is Mycena perlae, and this is the brightest glowing mushroom that I've ever seen. And so when I found this, I got hypothermia just photographing it because it was so much fun to photograph. But this is only a 6-second exposure with pretty low ISO, so really bright, and it's fun to just take a cap and throw it because it looks like a shooting star. And it's only the caps that glow. And so my friend has named it after his girlfriend, Perla, which is not a good idea because they were broken up before it even got published. So it's usually best to name a mushroom after a unique characteristic that it has, because in 100,000 years, people will still be using that name. And if you just name it after your friend, the name won't really have much meaning. Yep. Yep.
When it comes to naming mushrooms, how do you know that they haven't been named or that people haven't had historical discussions about what they call them, but maybe haven't been written down, maybe local groups in different parts of the world, right? And somehow the legitimacy comes from being named in English or being named in a different context by someone.
So to name a mushroom, it has to be validly published. So there's rules on how they get validly published. So all of the mushrooms that are undiscovered, people have been seeing them for not millions of years because there haven't been people for millions of years, but people have been seeing them for at least 10,000 years, 100,000 years, and some of them they have names for, others they don't. But until it's validly published, that's just people talking, and so.
What does it mean to validly publish?
So there's a thing called the International Code of Botanical Nomenclature. And so if you want to name something, you have to follow the rules in this International Code of Botanical Nomenclature. And they have rules like you can't name it after yourself, and it has to be like derived from Latin or Greek, and you have to have a type specimen, so that means you have to collect some of the mushroom and deposit it in a herbarium, so there's an official record. So if later someone's like, what is this mushroom, they can go to the herbarium and find your original collection, and they can study it. So that's a lot of what we're doing now is finding the holotypes, so it's called a holotype, is like the official collection, and then studying it and figuring out exactly what it is. And a lot of times people are wondering, these names are kind of ambiguous, and you have to find the holotypes and study them. And so until they get published with following the rules of nomenclature, then the name's not official. And then the first time the name is published officially, then that's the official name forever, and then people have to use it forever. And
scientifically, too, I think what you refer to is it's not getting a common English name, like, honey mushroom. It's getting an official scientific name in Latin or Greek, which is used internationally by scientists all over the world.
Yeah, and it's kind of nice because you see people, Chinese scientists, and you have no idea what they're saying because you might not know Chinese, but as soon as they start talking about the trees or mushrooms, they use the scientific names and you know exactly what they're talking about. The common names are there's a lot of people you say honey mushroom, but that refers to 50 different species of mushrooms. So if you want to be specific about which honey mushroom you saw, you have to use the scientific name. If you're just referring to any honey mushroom, you can use the common names. And then some people like to use the indigenous names, but in California we had 30 different indigenous languages, and they all had different names for the mushrooms. So then you say which indigenous name do you want to use? And you could use any of them, but usually the indigenous names refer to a whole bunch of different mushrooms. They might have one name for a whole bunch of different mycenas, and if you want to be specific about which mushroom you're talking about, the indigenous names won't work. But if you're talking about a chanterelle or something, then a common name or an indigenous name works just fine. But we have now we know we have 7 or 8 different species of chanterelle in California, and the indigenous people wouldn't have had different names for the different species of chanterelles. So a long time ago, they didn't really make these fine distinctions. They didn't use microscopy and DNA sequencing, so they didn't really zero in on all the different species that we had. But one reason that it's really good to name these things is because if you want to conserve it, you need to be able to talk about it. And so there's a lot of very rare mushrooms in California that only grow in one little tiny part of one forest. And so if you can name one of these really rare mushrooms and publish it, then you can say you shouldn't cut this forest down because this mushroom only grows in this little patch of forest. But if the mushroom doesn't have a name, then you can't really make that argument because there's nothing to call it. And there are so many undiscovered mushrooms. Even in California, you can just go out in your backyard and find undiscovered mushrooms. Here's one that didn't have a name when I found it, but now it's Mycena guzmanii. And this is another one where the leaves glow really bright. So this was named after Dr. Guzman, who named a lot of the psilocybes in Mexico. Here's another one that doesn't have a name. This one I found in Jalisco, so it makes the sticks glow really bright. And it has this off-center cap. And then this one, I have no idea what it is, but it's like a glowing bottle brush. And so I found this in southeastern Mexico, in Jalisco, in the cloud forest. And it was like up like 10 feet above the ground. I had to kind of like knock it off with a big stick, and then maybe one day I'll figure out what it is, maybe not. So glowing mushrooms, the bioluminescent mushrooms, they're pretty hard to photograph because they're super dim most of the time. So you need to use long exposure, although, now these days cell phones have pretty good night mode. So if it's a pretty bright glowing mushroom, you can use the night mode on your cell phone. If it's one of the more dim glowing mushrooms, then the night mode just won't cut it. But if there's the moon out, you can still see the glowing mushrooms when there's a moon, but then everything in the woods is glowing. So you really want to go out when there's no moon. So if the moon's out, you can wait till the moon sets or go out before the moon rises, or just go when there's a new moon. And when there's moonlight, it's like every white piece of bark, every piece of trash, it all is glowing. And then when there's no light at all, then only the bioluminescent stuff's glowing, and you can just walk through the woods, and anything you see glowing is either a glowing insect or a glowing mushroom. And it's really dangerous to walk around the woods at night, so it's nice to have like a good trail so you don't walk off a cliff. And you should wear glasses, poke your eye out. And if there's a city nearby, then you can still do this, but you have to go on a very clear night. So if on a very clear night, all the light from the city goes straight up into space, and it's still very dark, even near the city, but on a cloudy night, it just bounces right back down off the clouds, and everything looks like it's glowing. And this focus stacking is really nice for bioluminescent photography, because then you can keep your aperture all the way open, which really helps not needing super long shutter speeds. And then the other kind of glowing mushrooms are fluorescent mushrooms. So these are mushrooms where you shine a black light, and then what they're doing is they're converting ultraviolet light into visible light. So ultraviolet light has very high energy. It goes a shorter wavelength, and then the light that you can see, visible light, has a little bit less energy. So there's hundreds of thousands of different chemicals in nature where when you shine higher energy light on them, they will take that light and release lower energy light, and that we perceive as different colors. So this is Entacebe or Entoloma trachyaspera, and this one I found about a month ago in Mendocino County. And by the way, the mushroom season is really good in Mendocino County right now, so you should definitely go there if you want to find lots of good mushrooms. Although I think it's also really good, like Fort Bragg, or not, from Santa Barbara to like Big Sur. So you probably don't have to go that far. But there was mushrooms everywhere in any case. And here's a Cortinarius fuscoflavidus. So this one was just named a couple years ago, and it has this really dark cap. It's from the leprosity section of Cortinarius, and these are super bright. So you shine a black light on them, and you can see them from like 100 meters away. They're ridiculously bright. And I just figured out what this mushroom was like 2 days ago, because Hart Singer found the same mushroom, and he did the DNA sequencing on it, and then uploaded the DNA sequence to iNaturalist. So I searched iNaturalist for Cortinarius that had DNA sequences, and I'm like, oh wow, there's that super bright fluorescent Cortinarius. And when I checked the DNA sequence that had just been discovered a couple of years ago, Cortinarius fuscoflavidus, and it matched the holotype 100%. So that was discovered in Washington, so it turns up in Washington, California, Oregon. But really cool looking thing. And this one is Tricholoma aurantiolavaceum. And I found this one at Prairie Creek Redwoods. So almost California coast, almost up to Oregon. And this one, I took the ultraviolet light and shined the light down from the top. And I shined it for 6 seconds for the one on the left and 6 seconds for the one on the right, so it looks like it's glowing from the inside. But the ultraviolet light that I use like this kind of light here. And this light is about $100, and it's a 365 nanometer light. Although BAT discovered recently that there is a $35 light that works just as well. So I'll show you that link in a little bit. But they're really nice, makes it so much fun to walk around the woods at night. And so you can just like here's a rusula here, and you can see it really makes rusulas light up really well. And I also like this Entoloma. This is Entoloma fruginans, and it's only lighting up right at the stem apex, and it's a beautiful gold color. So you get all sorts of different colors, and it's just so much fun to walk around the woods with one of these black lights, because it's like a whole new world, and a lot of flowers are ridiculously beautiful. A lot of plants are really beautiful. Some of the insects are also really beautiful, and a lot of the mushrooms as well. But here is a Galerina. And then here's this Bovista, a little puffball from Telluride. And this one I like a lot. I found it about 3 months ago in Veracruz, Mexico. And so this is southeastern Mexico. And this one is Psilocybe subtropicalis. So Psilocybe subtropicalis is one of the psilocybin mushrooms, and it's closely related to Psilocybe zapotecorum. And looks really cool in ultraviolet because the gill edges are fluorescent. And that's probably due to the presence of beta-carbolines. So beta-carbolines are those MAO inhibitors that they have in ayahuasca, so it causes the DMT to be orally active. But they've recently discovered that they're also in psilocybin mushrooms. So who knows if they're there in enough quantity to change the psychoactive effects, probably. But in any case, you can see it with an ultraviolet light. Another one, here's one I found this summer in West Virginia. This is Loradiomyces squamosus. This one's pretty rare in California, but it does turn up here. And it has really cool looking gill edges. And then just like zooming in all the way, you can see the gills here. And then here's some honey mushroom rhizomorphs from Amsterdam. And these have some algae on them. And the algae has chlorophyll, and the chlorophyll fluoresces red. So that's why these are like that. But yeah, it's honey mushroom rhizomorphs. Looks like black bootlaces. And here's the Cordyceps militaris. These were grown by Ryan Paul Gates. And he does a lot of Cordyceps militaris breeding. So here's how they look in regular light. And then in ultraviolet light, they look really cool. And here's a picture of me taking that picture. So this was at MycoFest. And so that's a mushroom festival run by William Padilla Brown out in Pennsylvania. Really cool one to go to. I'll probably be there this year too. And then here is a Selaginella. And so Selaginellas are these kind of ancient living fossil type of mosses. And so they look really cool in ultraviolet light. And so I just took a picture in regular white light too. But they have these waxes, and the waxes convert ultraviolet light into blue light, so it gives it protection from the sun. But then this freshly growing tips don't have those waxes yet, so the freshly growing tips have chlorophyll that is fluorescing red. And then here's a redwood rooter, Coleorhiza umbellata. And these are both the same mushroom, but when they're really young, they fluoresce bright blue, and then when they're really old, they fluoresce like a sea green color. And then here's Viola glabella, a violet. And so most plants will fluoresce either not at all because they have UV protective waxes, or they will fluoresce red because of the chlorophyll. But these ones happen to fluoresce blue. I have no idea why, but it's very unusual. I find these at point rays. And so the top of the leaf fluoresces blue, and the bottom of the leaf fluoresces red. And here's something from the Apiaceae, the celery family, maybe poison hemlock. And here's some spruce tips. So the very fresh spruce tips are fluorescent blue. And then after a while, they're fluorescent green. And here's Albogenopolis nanus. And Albogenopolis is a new genus that has not been discovered yet, but maybe it will soon. Hasn't been published yet. It's in mushrooms of the Red Blue Coast. It's kind of like a Gymnopilus, but it has a really viscid cap. And they're super fragile. So all the Gymnopilus, they have very dry caps. So I sequenced this one. It came out like off to the side of Gymnopilus, so I think a different genus sounds like a good thing to use for it. But this one, I put KOH on it, and it totally changed the color of the fluorescence. So without potassium hydroxide, it fluoresces green, and then with potassium hydroxide, it fluoresces blue. And this is really common, like in Cortinarius, where you add potassium hydroxide, and it changes the color of the fluorescence. Here's Chimonophyllum. So it's kind of like a little oysterling type thing from Mount Rainier. And here's some blue chanterelles. So these I found in Mount Hood, Oregon. And so in white light, they look like black chanterelles. And then they're like nice blue and ultraviolet. Here's Pholiota spumosa, a really common Pholiota that you see around here. They look just like sulfur tufts, except they have lighter brown spores. Here's some that I found in Michigan. And some from Bay Area. And here's a Cortinarius. I like this one because it kind of looks like a tiger's eye. And here's Meadomyces dissimulans. This one looks just like a grosbeak, but it does not smell or taste like a grosbeak, so no cucumber odor. These are actually really common, like on the sides of trails, but nobody ever really notices them because I think it's just another grosbeak. And then these spots, I find them on leaves. I have no idea what causes them, but you can see that they're kind of like reddish spots, and then with ultraviolet, they're bright orange. There's also some that are bright yellow. So these I photographed in Michigan, though I've also seen this in Washington. And so I used a microscope to try to figure out what these were, and I couldn't see anything, just plant cells with the microscope, so never figured it out. Here's some Ganoderma. This one is Ganoderma lobatum. Here's another Pholiota. Some hedgehogs, Hydnum. Here's some Hemimycena. And the most popular and common fluorescent mushroom is Hyphiloma fasciculare. And so this one is the sulfur tuft, and they are super bright. You find them like 100 meters away if you have a good light. And here's also some of this mysterious orange spots on the leaf. These I photographed up in Washington. And then with the microscope, you can see like which part of the mushrooms are fluorescent. So this is Hyphiloma fasciculare. And on the left, you have the lamellar trema, which is like the inside of the gill. And on the right, you have the basidia and cystidia. So the lamellar trema fluoresces yellow, and then the basidia and cystidia fluoresce green.
Wait, did you change the bulb on your?
No, I used for this one, I used a Zeiss fluorescent microscope. So they're pretty expensive, and they have like their own ultraviolet source. But you can just change the bulb. If you're not trying to do really high resolution, you can just shine a really bright ultraviolet light at the stage, and it works. It works fine at like 40 times or 100 times, but you try to do 400 times, it doesn't work very well. But what you can also do is like get like maybe a 10 watt or 50 watt 365 nanometer LED, and just put that where your bulb goes, and then you would have a fluorescence microscope. You know, probably save about $10,000 on it. But then your scope is a little dangerous because it's like sending ultraviolet at you. But you know, when you magnify 400 times, it also makes it 400 times dimmer. So it's probably not that dangerous, but it'd be a really good idea just to connect a camera to your microscope so you're just looking at a picture instead of looking directly into your ultraviolet light. Why is there a black spot? Oh, that's the pointer. So some fancy microscopes have a pointer, so you can like kind of point at what you're looking at, and this one just happened to have a pointer. A lot of Hyphilomas are really fluorescent, all of them I think. So this is Hyphiloma lateridium, the really popular edible Hyphiloma from out east. And then the Septotrauma gives you nice like purple colors. That I found in Texas. And then here is Callistosporium pseudophyllum, which is a species that was discovered just a couple years ago. And they are super bright, and they're all over Texas. Here's some Russula from Florida. Here's some Russula from California. So these have a really nice electric blue cap. And then with white light, you can see it's like, you know, a nice red cap. So this one was Russula zelleri, which grows with spruce. There's another Russula from Florida. And then a lot of insects are really cool too. So this is from Big Thicket, Texas, and it has fluorescent legs. And they're super bright, so you can imagine just like what it looks like when it's walking. It looks amazing. And so if you find something like this, definitely shoot a cell phone video. And then the scorpions are always super fluorescent. So this is the Centauroides, which are the really dangerous Arizona bark scorpions, little tiny ones. And then here's one I found at the Alabama Mushroom Festival, just a couple months back. And then there's a ton of fluorescent lichens. And one of the coolest things about the lichens is that you see a tree full of lichen, and there's like it's all green, but then when you shine the black light on it, certain parts glow really brightly, and then you shine the white light, and you see like the whole tree is green. So it really helps you separate out the species of lichen. So this one is Pyxine subcinerea. And this was at the Namaphore a few months back in Missouri. You get all sorts of different colors out of lichens. So this is a yellow lichen that I found in Amsterdam, and then turns bright orange in ultraviolet. And then this one's really common. This is Ochroleucia. And so it has these like cool little cups. It looks like bright orange spray paint. And again, like when you're with white light, you can't see very much at all. If you look really close, you can see the difference, but it really shows up in ultraviolet. Here's Achirospora, really common lichen out in the desert. And then here is a lichen. This one is like Tuckermanopsis. And so I took this photo right at sunset, and I used a wide-angle macro lens, so you get like really good detail on the lichen, but you can also see the sunset in the background. And then with white light, looks like that. Yeah, I put all my photos on iNaturalist. And they're also all on Mushroom Observer. But iNaturalist reduces all of the photos to about 2.6 megapixels, and then Mushroom Observer keeps the full 45 megapixel photos. So if you want to like search for these photos and print any of them out, definitely search on Mushroom Observer. But you can find them on both sites. This lichen I like a lot because it looks like a jellyfish. It's really common on coastal California, like Mendocino County and Humboldt County. And then this one is Usnea. And then here's some bacteria. And the bacteria is on a polypore up at Mount Rainier. And so in white light, you can see they're just these bright pink spots. And then with ultraviolet, they really glow. So ultraviolet photography is so much easier than bioluminescent photography. And the reason is because these ultraviolet lights are really bright. And so you're not trying to capture like really tiny amounts of light. Like these things are really bright. So you can just take cell phone photos. And the cell phone photos turn out spectacular a lot of the time. The most important thing with a cell phone is that you turn down the exposure compensation. So what you do is you like touch the subject to focus on it, and then like on an iPhone, you'll see that little sun show up, and you drag the sun down. And that will turn it from kind of like a regular boring photo to like a super awesome looking photo. And with Android, same thing, you just like kind of focus on it, and then there's like little sliders on the side, and you just turn the exposure down until it looks really awesome. And then there's two kinds of black lights that are really common. There's 395 and 365. So if you've ever been to a black light party, that's 395. And those lights give you a lot of purple tones. So if you ever see ultraviolet photos that have a purple background, that's 395. The 365 is further outside of what your eye can see, because your eye can see right about down to 390. So 365 is visible to your eye, so you don't get those purple tones, but also a lot more stuff shows up in 365. So the 365 lights are a little bit more expensive, but they're totally worth it. So you really want to avoid the purple lights. The ones that look kind of bluish are the 365. And it's really important to get a real bright one, because the dim ones are just not very spectacular, whereas a really bright one, especially if it has a good beam, you can kind of like shine it off in the distance or kind of from a lot further away. And so there's a bunch of different lights that are good, but I guess the best one is that $35 one that Bat found. And I will show you that. And if you message me, I can just link you to the Amazon thing. So I use a DSLR camera with a macro lens to do UV photography, because then I can control the exposure really well and get really good detail. But it works so well with a cell phone that it's really not worth the effort to set up your tripod and everything. And then here is the electromagnetic spectrum. And so the stuff on the right is pretty safe. And that's like radio waves and cell phones and microwaves and infrared. And then you have the visible light band. And so infrared on the right, and then it turns, then you get red light around 700 nanometers. About 530 is the bioluminescent mushrooms. And then it starts to get dangerous to the left. So purple light is still pretty safe, and the ultraviolet that we use for this is still pretty safe, though I wouldn't stare into it. But once you get into the shortwave ultraviolet, then it starts to mutate DNA. So it gives you skin cancer and sunburn really fast. So I have some really dangerous ultraviolet lights that are different than the ultraviolet lights that I use for photography. And those, when I use them, I have to put on sunscreen, like all over my face and hands before I use them, and wear glasses, because otherwise they'll give you cancer and sunburn real quick, and they're really good for sterilizing stuff. And then if you go even shorter, you get X-rays, which give you cancer, and gamma rays.
Is that UVA and UVB?
Yeah.
Okay.
Yeah.
Are those more powerful lights better for fluorescent mushroom hunting?
No. I ordered one that was, so my favorite wavelength is 365. So the 365 is not particularly dangerous. I accidentally shine it in my eye all the time, and that's probably bad, but it's not that bad. The more powerful ones are like 254. That's what you got to wear sunscreen for. And I tried it one time, and I was really disappointed with the results. Like, just not very much was fluorescing, so it's not worth like having to cover up with sunscreen, and it just didn't make stuff fluoresce very well at all. So yeah, they're not very good, even though the one I bought was a super powerful one. They are really good for minerals. So the more dangerous lights are really good for like certain rocks, but for mushrooms, not so much. Any other questions? Oh, I guess I have a couple more slides, and then I'll take questions. So the good UV lights, this is like the $100 one right here. This, this one here, $100. But it's just as good as the $35 one that found. So you probably want to get that one instead. But if you want something that you can just order off of Amazon and have it arrive before Christmas, then get this UV Beast V3. Those things are about $80, and they're pretty good. Also, if you like glowing stuff, you probably like the Fluorescent Forest Facebook group. There's a bunch of different photographers that post their glowing stuff on there. And you can see all my glowing photos on there. And it's a cool place just to hang out and share glowing photos. And then there's also an iNaturalist project for glowing things. So there's one called UV Fluorescent Organisms. So whenever I find anything that's fluorescent, even if it's just like my friend's dog, which is fluorescent, by the way, like white dogs are fluorescent. Anyways, I just add it to that project, and then you can keep track of all the fluorescent things. And they've found about 1,100 different fluorescent species, and they're adding more every day. So there's a lot of fluorescent stuff out there. There's also one called bioluminescent organisms, and those are the ones that glow in the dark. And there's a whole lot less. I think there's more like 150 species that glow in the dark without ultraviolet. But if you find anything that glows in the dark, definitely put it on iNaturalist and put it in the the Bioluminescent Organisms Project. And if you have any more questions, I can take them now, or you can contact me here. Thank you very much. Yes.
Hi, I was just wondering how you got into becoming a mycologist.
How did I get into becoming a mycologist? Oh man, that's a good question. I guess it was like 2001, and I moved to California because I grew up in Chicago, and I moved to California. And when I was in Chicago, I had a lot of friends, and they sold a lot of drugs. And then I moved to California, and I didn't know any drug dealers at all. So I just started looking for mushrooms. And I did it exactly the wrong way, and I don't recommend this, or maybe I do, but if, what I did is I just started walking around looking at all mushrooms, just hoping that some of them would be magic. And I finally found magic mushrooms like 2007, so it took me like 6-7 years to find them. What I should have just done is just Google like where do magic mushrooms grow, and I would have found them in like 3 days that way. But by the time I found the magic mushrooms, I was just like really hooked on mycology. And so around 2010, I quit my jobs just to do mushroom stuff full time and try to make a living as a mycologist, and that's what I've been doing ever since. Yep.
Is that snake in the photo in UV light or is it?
No, that's just a regular light. That's just a regular snake. But I thought it looked cool, and so I threw it in here. But some snakes do look pretty cool in ultraviolet. But I took this photo in the middle of the day, so it would have been kind of hard to get ultraviolet. Yep.
Including setup, how long does it take any given photo?
Including setup, well, I use a tripod for everything. And so it's maybe 15 seconds to set up a tripod, 15 seconds. But to set up the mushrooms, like usually I'll set the mushrooms up for a while. Like you can see there's not just one mushroom, but like I'll find like a good patch of mushrooms. So I would say I'm sitting down for like usually on average about 5 minutes. And then as much time as I spend sitting down is about how much time it takes on the computer. So I usually probably spend 5 minutes out in the woods and 5 minutes on the computer, so about 10 minutes. And so that's one benefit of a cell phone, because with a cell phone, the photo is instant. It does all the post-processing for you, and so it's just a couple seconds. So cell phones are really cool in that way, but I think, I really like just like sitting down and spending a lot of time with the mushrooms. It's really relaxing and nice, and then the quality of the photos is really good. When you have a tripod, you can keep taking the photo over and over until it comes out the way you like it. So I'll take the photo, but maybe it'll be like blurry, and I'll focus better, and then I'll notice there's like grass in front of the mushroom, so I'll take the grass away and just keep taking better and better photos until it's perfect. So sometimes I like to spend a really long time with them, like when I go out with Alison Polak. She's a very famous macro photographer. And when I go out with Alison, we'll just sit down in front of a log and spend 4 hours sitting in front of a log, taking the log apart. And after 4 hours, we will have photographed like 10% of what's on that log. But it's so much fun, and like the photos just come out like super beautiful. So definitely worth it. But when the rains are good, I'll usually go about 20 hours per mile. And then, I'll go out all day. And when the sun sets and it's time to go back to my car, it's only like 50 feet away. When the rains are bad, then I'll walk like 10 miles. Yes?
Hi, Alan. We had a speaker a few years ago talking about bioluminescent mushrooms. You mentioned you weren't sure why mushrooms bioluminescent.
Was that Brian Perry?
I think it might have been. Yeah.
Probably. That guy's awesome.
Go back and look through my old notes. But his theory was that in tropical, most of the bioluminescent mushrooms are found in tropical climates, and not typically in more temperate or subarctic climates. Anyway, because it's so hot all the time, they don't have a good way to release excess metabolic energy through heat, because the ambient temperature is so warm, so they release it through light.
Yeah, that's definitely Brian Perry. And that guy is super cool. I went mushroom hunting with him a couple days ago. And so, yeah, that's definitely possible. And also that chemical reaction that releases light, it also takes oxygen. So if they have like kind of like free radicals or extra oxygen that they want to get rid of, maybe they're kind of using that chemical reaction to get rid of the light or get rid of the oxygen. That's definitely possible. Though, you know, most chemicals that mushrooms produce, we don't know why they produce them. There's a new paper that came out the other day that found that psilocybe cubensis has 1,400 different chemicals in it. And I think any mushroom you analyze would be very similar. And they're totally different chemicals, so maybe we only know what like 2 or 3 of those chemicals do. And so it's kind of like you pick any chemical the mushrooms make, and it's very difficult to figure out why they make it. And we can speculate, and there's a few different, you can guess, but it's really hard to be sure. But that's definitely possible. He could be right. Yes?
Are there a lot of folklore explanations for bioluminescent?
They definitely reported them a long time ago, like hundreds of years ago, maybe even thousands of years ago. They would talk about them. And yeah, you know, there's some in Europe. And so, yeah, they mention them. Yep.
Do you ever do anything like in terms of institute work where you're doing gas chromatography or mass spectroscopy? Like, I mean, you're working in an awesome work with people, you know, like as your lab. Do you have anything you can do right there on the spot that you kind of figured out works for you?
I would love to get one of those. I really want a gas chromatography mass spec machine for my living room, so I can just take all my mushrooms and just feed them through and see the chemical profile. But the machine I want is about $250,000, so I'm saving my pennies. And I'm not a chemist. Like, I like chemistry, and it's my hobby, but I'll never be very good at it. So I have a lot of chemist friends who do this stuff. And I would really like to kind of like correlate the DNA sequences with the mass spec data and kind of like do more of that stuff. But I haven't done any of that myself. Yep.
Have you ever read the book Holy Mushroom, Holy Cross?
I've heard of it, but I haven't read it.
Okay.
I probably wouldn't like it. Yes. There are no good field guides for Mexico. So what you should do is take pictures of all the mushrooms you find and put them on iNaturalist. And if nobody identifies them, then just message me and I'll go through your iNaturalist account and identify them. There are some very bad field guides for Mexico, but it's not worth your time to even get them.
To get started, to know where to go and when.
So where to go? You should just search my iNaturalist, because I've been going to Mexico for like 15 years, and everything that I find, I photograph and put on iNaturalist with exact coordinates. So if there's a certain thing you're looking for, you can just search my account for that genus, or if you're looking for glowing stuff, just search my account for the things in the Bioluminescent Organisms project, and it'll give you exact GPS coordinates where I found all that stuff. And then just kind of like, probably go exactly there, and then just look on the satellite view for similar habitats, like in the next mountain over, and you'll find all sorts of cool places to go. Mm-hmm. Yep. Can you cross Psilocybe septicorum with Psilocybe subtropicalis? That's a really good question. And so they're different species, but they're very closely related. So a lot of times if you're trying to like cross different species, what I like to do is build a phylogenetic tree of the genus. And it kind of has this tree of life where you can see that the more closely related species are closer on the tree. And the things that are more close are more likely to cross. So if it's like the same species, they might cross like 75% of the time. And then if it's like a different species, it might cross like 10% of the time or 1% of the time. So you might have to do it on like 100 Petri dishes. I don't know if anyone's ever tried it. I think if anyone has, it'd be this guy, Kyler Diamond. Yeah, you should ask him. I think he might have done it. Or maybe Julia Matucci. I think I saw on Facebook one of those people had tried it. I think it was that one. Because they're definitely crossing Psilocybe cubensis with natalensis, which are close sister species. I think it would be a good idea to try to cross those because they are really close and they're both super potent.
just go out at night?
I do both. What I will do is go out, and whenever I see a mycena, I'll photograph it and throw it in my tackle box and save all the mycenas, because most of the glowing mushrooms are in mycena. And so then just bring the tackle box either into a super dark room or just wait for night. And then I'll take a photo of the tackle box with a really long exposure and to see which compartments are glowing. And if I'm collecting like 10, 20 species of mycena, I'll always see at least one glow, because most mycenas will glow like at least a little bit at the stem base. But then also just walking around in the complete darkness is a really good way to just be able to spot them at a distance. So I do both. Mm-hmm. Yep.
You mentioned earlier the potassium oxide changing the colors.
Mm-hmm.
Is there a particular reason?
Let's see, is there a reason that potassium hydroxide changes the colors? Well, potassium hydroxide is the world's strongest base, so it changes a lot of things, and that's kind of like why we use it. So we put that on just mushrooms for identification purposes, and a lot of the pigments are different colors at different pHs. So you just put it on mushrooms, and depending on the color change, that gives you taxonomic information about what species it might be. Just happens that a lot of the ultraviolet pigments also change the way that they fluoresce when the pH changes. So that's the reason, and it's pretty unpredictable. Like, you never really know what will look cool in ultraviolet light until you shine it, or what looks cool in KOH and ultraviolet light until you try it. So it's kind of fun, just to like try every mushroom on the table at a fungus fair or something. It's like thousands of specific reactions. So there's thousands of pigment chemicals in mushrooms, and some of them are different colors at different pHs. And then there's thousands of fluorescent mushrooms and different fluorescent chemicals in mushrooms. And some of them are the same at different pHs, and some of them are different. So there's just the chemistry of fungi is extremely diverse. And any mushroom you pick up off the table will have over 1,000 chemicals in it. And each mushroom has different chemicals. That's why some mushrooms are poisonous, because they just have some chemicals we don't get along with. Yep.
Are there any mushrooms with, or have you found any mushrooms with luminescent spores?
I have not found any mushrooms with glowing spores. I did find a really cool fluorescent spore print, though. Let me pull it up for you really quick. Before I do that, let me show you this. This is the ultraviolet light that's only $34. And this is just as good as my $100 light. And so what you should look for in a good ultraviolet flashlight is the wavelength of 365. So this is not the purple toy, but an actual serious black light. And then also, if you look at the front, see how the front looks black? So these LEDs, the 365 nanometer LEDs, they make a little bit of light in the 530 nanometer range, which is like aqua. And that's not good, because you want pure black light. So they put this black filter, and this filter blocks the visible light, so you get like nice pure black light. Thank you so much. Oh, that's awesome. Thank you. He says, that was cool. It's adorable. But let me show you really quick this photo. So from the fungal microscopy Facebook group, it's a really cool group for mushroom microscopy. But the cover photo, this is just a picture that I took in my garage a couple weeks ago with my cell phone. But kind of a cool picture. And so this is Entoloma subcarneum, which is a beautiful mushroom. But it had this awesome fluorescent spore print. But I think that the spore print isn't actually fluorescent, because it's only on the edges. So I think that some of the fluorescent chemical from the mushroom leaked into the spore print and wicked across it, like chromatography sort of. But I'll show you this mushroom. I should have put it in this talk, because it's awesome. Entoloma subcarneum, one of the world's most beautiful mushrooms. This one I found like a month ago in Mendocino County. It looks like that. And then with white light, it looks like this. Yeah, so this is the one that had the fluorescent spore print. Any other questions? Yep.
Are
do you have any recommendations for what mushrooms we should be looking for in SoCal, or where we should be looking?
Oh man, there's so many good mushrooms, but there's not a lot of work that's been done with coast live oak mushrooms, so it's a really good tree, and so many of the mushrooms that we find are undiscovered. Especially what needs a lot of work is Russula. So most of the Russulas you find, they don't have any names yet, and that'd be a really cool thing to name. I was just spending a week hanging out with Bart Buick, and he lives in Paris, and he is one of the world experts on Russulas, and he also named a lot of our chanterelles. So he taught me how to make really good Russula collections. Basically, you just take good pictures of them, but also cut it in half. So take a picture of a cross section, and also you have to make a spore print. And then once in the morning, once you have the spore print, you have to scrape the spores up into a pile and take a picture of that to get the right color. And then just mail it to him and he'll name them. So that would be a really cool thing to do, is to collect the Russulas.
That box that I gave you, I think is Russulas.
Yeah. Probably a new species of Russula here.
I think they glow.
They definitely glow, yeah.
The caps are.
And pretty much all Russulas glow. And that's just something. Oh, awesome. Here's, Bat gave me, brought up this nice ultraviolet light. So here is the $34 ultraviolet light with the Russula. And then here's the $100 ultraviolet light. And you can see that, if anything, the $34 one is better. So.
Did you see the cap though?
Oh, let's check the cap box. Oh, that's beautiful. It's like an electric pink tie-dye.
There's like green and the younger, I don't know if they're younger or different.
Oh man, that one's even better.
Yeah.
Yeah, that's beautiful. Yeah, Russulas are really good. Yep.
Alan, you mentioned that Russula is really fully described in Southern California, or really fully documented.
Yeah.
But I can say the same for Portland area.
Yeah, but there's a lot more people working on Cortinarius. There's Michelle Torres Grant is collecting a lot of Cortinarius. And whenever I find a Cortinarius in Southern California, I sequence it, and then I message Dimitar, who's our expert on California Cortinarius. And he always says, oh yes, I know that one. It doesn't have a name yet, but I'm working on it. And then when a year or two, it'll have a name. And he's right, he does publish a lot of new names. So all of the Cortinarius around here, except a couple of the really rare ones, they're not really that mysterious. People know about them and they're thinking about them and they're working on publishing them. The Russulas, on the other hand, nobody's thinking about them, no one's publishing them, but they're very common. Even the most common Russulas that you see 100 times a day, nobody's working on them, and we really should. I've sequenced a few Russulas from Southern California, and they don't really have any GenBank matches, hardly at all. People just aren't working on them.
This is a Cortinarius.
And Cortinarius can be beautiful in black light too. Oh yeah, this Cortinarius is awesome. You can see that most of what's fluorescent is the stem base. And so the stem base is glowing like a yellow-orange, which is really cool. You have a question?
What was the name of that guy that you said that was in Paris?
What's that?
The guy that you said?
Oh, his name is Bart Buick. Byuck. And if you message me or email me, I can give you his email address. Or you can probably just pull up one of his papers. But he lives in Belgium. He works in Paris. And he's a very good taxonomist. He's super serious dude. And so, yeah, you can definitely mail him Russula. He would love good collections, but make sure you make spore prints and make good collections, because he needs good collections.
So since I didn't cut those, will they not work for that?
I mean, you'll still get a good DNA sequence off of it. You can still use microscopy. He won't name anything off of one collection. He wants at least a minimum of 2 collections to name something, so he knows that he's getting a good representative sample. So, I mean, this could be one of the collections, and then maybe the other collection would be the really good one. So I think it's still useful. But
I did make a sport print.
oh, awesome! Yeah, that's great. But yeah, I found 2 Russulas today. One of them I cut in half, and it had all these chambers in the stem. And the other one I cut in half, and it had a solid stem. And that's what we're looking for, is if the stem is chambered or solid. Well, thank you very much for coming.
Say it again,
Entoloma subcarneum.
subcarneum.
Yeah, and they actually grow pretty far south. Like, you can definitely find them in Big Sur and maybe Santa Barbara.
Like,
Yeah.
so just coastal, not inland.
Yes, not inland, only coastal.
Definitely wet, damp.
I think they only grow with redwoods. So,
In the redwood?
in the redwoods.
Interesting.
Yeah. I've always found them in redwoods. Oh, also cypress, Monterey cypress. They grow cypress.
Cypress and redwood is okay. We have two big redwoods in
I'm gonna be fruiting the Zapspores you gave me this month.
Oh, that's exciting.
You gifted me Zapspores in Oakland.
Oh, cool.
Yeah, man, it's going really well.
Yeah, those are some of the best fruits. I think it's totally worth the extra time that it takes.
Yeah, and so far I'm finding that high nutrient substrate is prolifically. Versus
Yeah.
like standard card.
Yeah, I mean, Jordan Jacobs is doing really good with coconut coir and grain. But I still need to think they're not really that picky about humidity. So you just have to keep the humidity up really high. Because if you let the humidity go down, then some mushrooms don't really mind, but those get really angry. Hi.